It's predicted from Cancer Research UK's data that 1 in 2 of us will actually develop some form of cancer in our lifetime. Cancer itself can be very challenging to treat. There are certain types of cancer which have got good treatments with decent outcomes. However, unfortunately, there are other cancer types which have very low survival rates, and for those patients we need to do better. So my name's Dr. Tim Witney and I lead a cancer imaging research group here at King's College London. In my group we're really interested in imaging cancer itself, specifically trying to image therapy resistance in cancer. And the reason why we're so passionate about this area is because if you can pick out those patients that are not going to respond to, say, chemotherapy or radiotherapy, you might be able to offer them some alternatives that might improve their survivals. And in order for us to get something that can be tested in humans, we need to make sure that this is working properly. And to do that, unfortunately, sometimes it's necessary to grow tumours up in animal models, for example, in mice.
The mouse provides a really good model organism for us to study cancer because that's our only way of seeing really how tumours grow and respond to treatment in a physiological system where you've got all sorts of factors which are going to impact tumour growth and how it responds to treatment. I'm Maddie, I'm a research assistant here in the Whitney lab in the School of Biomedical Engineering and Imaging Sciences at King's College London.
So there's no perfect model, right? In order to try and get as close to the human condition as possible, we take a range of different models and put them together and see whether we have enough evidence to try and take these into clinical trials. So within my group, we're trying to innovate new alternative animal models of cancer to replace or at least reduce the number of mice that we end up using. And one of those is the fertilised chicken egg. What we found is that we can get very similar data by using a fertilised chicken egg as we can with the mouse.
Within the chicken egg you have the growing chicken embryo and the yolk sac, and surrounding that is the chorioallantoic membrane, or CAM as we call it. And that membrane is highly vascularized, which means there's a good supply of blood vessels, which makes it a really good surface to be able to grow tumours on. And then we can study how they grow and how they respond to treatment within that 14-day period that we grow the tumours for.
So it's been shown actually up to that embryonic day 14 that the embryo itself doesn't experience any form of pain. So that's the whole reason why we would never use the embryo over day 14.
We start by keeping the eggs at around 12 degrees and then we move them to a humidified incubator at 37 degrees. And once they are moved to 37 degrees, that becomes embryonic day 0. And after 3 days, we take them out and make a window in the eggshell. At embryonic day 7, we remove the eggs from the incubator again and inoculate tumour cells onto the CAM surface. We then have a 7-day period to study how that tumour grows and potentially look at longitudinal treatments over that time. So this is an egg at embryonic day 14. So you can see the yolk sac is this yellow section on the right, and then the embryo is here. And over the top of it all is that chorioallantoic membrane. which has got these blood vessels all feeding into this tumour, which we inoculated at day 7, which is this white patch here. So now we can cannulate these blood vessels and inject our radiotracer and use this egg for PET imaging. For the PET imaging, we inject the eggs with the radionuclide and we then put them into the PET scanner. So we'll then get a signal as to where the radionuclide has been taken up by both the tumour and the rest of the organs within the growing embryo, and we can compare the tumour uptake and retention of the radiotracer that we're studying.
So what we see here is a 3D view of the fertilised chicken egg after we've injected one of our imaging agents. And the tumour that's rotating just here is lighting up, and the rest of this is the growing embryo itself.
If you scroll through here, this is like... We've seen so far that just like we can in mice, we can also grow and image tumours in eggs. We don't think this model is ready to replace mice altogether, but it provides a really helpful intermediate between in vitro and in vivo studies that could really reduce the number of mice that are needed for animal research.
So the question of whether we can fully eliminate animal models of cancer is a big one within our community. I don't think even with AI or with all kinds of intricate in vitro models we could ever recapitulate the complexity of the human body. So unfortunately, I think for a long time, at least within my lifetime, we will still be using animal models of cancer. For me, it's clear that we're in a very privileged position where we have the ability to work with animals and also potentially, end an animal's life. So I think we're obligated morally to see if there are ways in which we can reduce the number of animals that we use in our scientific research, and that we can still get to the same endpoint, which ultimately is clinical trials and hopefully human benefit, but with the lowest number of animals used possible.